Okay, in this video tutorial I'm going to talk about polysaccharides. So polysaccharides, poly means many, and saccharide stems from, I think it's the Latin, for sweet or sugar. So polysaccharides literally means many sweets or many sugars. And that's essentially all they are. Polysaccharides are a long chain of many sugar molecules linked together. So remember, from my last video tutorial, a monosaccharide is a single sugar, a disaccharide is two sugars linked together, polysaccharide just many linked together. So monosaccharides and disaccharides are considered sugars, whereas a polysaccharide is actually no longer considered a sugar. So poly also is because this is a polymer. It's a polymer. Now guys, if you study chemistry, you'll know what that term means. Poly means many, mer kind of unit, so many units pieced together. So DNA, for instance, is a polymer of nucleotides. It's made out of lots of nucleotides pieced together. Okay, so these things are not sugars. Alright, so that's worth noting. Polysaccharides are not, even though they're made out of sugars, they're not considered sugars anymore. And they're less soluble. And that's a really important point. That's one of the reasons that we actually piece biological that organisms or biological systems will often piece together sugars into these polysaccharides, is that if we left them as sugars, in some organs or tissues there would be so much sugar that essentially the cell fluid would become quite syrupy. So the solubility would become too concentrated, and that concentration of sugar in the cell would actually affect cell function. For one, it would mean water would actually flow into the cell to try and balance the concentration between the inside and the outside. Now that's an idea called osmosis that we'll look at later, but essentially too much sugar in a cell actually starts to become toxic. So by linking it together, we actually get a lesser concentration, and often these chains are often less soluble, so we don't have as much dissolved in the cellular solution. It's not as concentrated, and that's one way of dealing with or keeping those sugars, which are a valuable source of energy, without them becoming toxic to the cell. So I guess it's that age-old rule that everything in moderation. Too much of anything can be a problem. And for cells, well, they want to keep that sugar. It's a valuable energy source, but keeping it as sugar, too many sugar molecules in the cell is actually toxic. Okay, so our three types of polysaccharides are these: starch, glycogen, and cellulose. So we're going to go through those one by one. So starch, glycogen, and cellulose. Firstly, starch. Now I've put in brackets there plants. Starch is basically a way for plants to store their sugars. So starch is really only found in plants. You'll notice this bond here. So this is from the condensation reaction, we've got this oxygen that links the sugar molecules. Now that bond that links the sugar molecules is actually called a glycosidic bond. So just be aware that condensation reaction that links sugar molecules together is called a glycosidic bond. Alright, so let's kick it off. Starch is actually a mixture of two molecules, amylose and amylopectin. So if we look at starch, it's actually got a component of these two things. There's a percentage of each. And I believe that depending on the source of the starch, some starches from some plants will have slightly different proportions of amylose and amylopectin. Very slightly different proportions of each of those. So, what is amylose? Let's start with that one. Amylose is simply a long chain of 1,4 linked alpha glucose. So it's alpha glucose linked in a long chain. What does the 1,4 bit mean? Well, I'm trying to kind of show that here with the molecule in the background itself. So up here, if we've got the oxygen, we treat that as like number 0, then carbon number 1. Alright, 2 would be down here, 3 and 4. So these are linked end to end in a nice long chain, so that's nice and simple. Alright, where the 1, the carbon at position 1, is where there is a linkage to the next one, and the carbon at position 4 is where there's a linkage to the previous sugar molecule. So they're linked end to end in a 1,4 kind of linkage or pattern. So you can see here the 1 of this glucose molecule here linked to the 4 of this glucose molecule here by that oxygen, that glycosidic bond, where we had that condensation reaction occur that links these two sugars together. Alright, these chains can curve or form long coiled springs. So it's just got to do with the nature of the bonding. It doesn't form a perfectly straight molecule, it's got a slight curvature to it. So if they're very long, you can actually get coiled spring-like structures. Amylose usually consists of several thousand glucose molecules. So it really is a case of, often we talk about monosaccharides being 1 sugar, disaccharides being 2, and then polysaccharides are often thousands of glucose molecules long. Amylopectin. Well, amylopectin is really quite simple. It's nothing too crazy. It's basically the same thing. So it's usually just a shorter chain, not quite as long, not quite those thousands of glucose linked end to end. But it also has branches. So what do I mean by branches? Well, the branches are formed by 1,6 linkages. So what is a 1,6 linkage? Well, basically what happens is carbon 6, the one that sticks up the top here, so remember oxygen would be here, 0, 1, 2, 3, 4, 5, 6. So guys, that's one way you know you're doing the counting right. If you're not sure where to start, you should always finish on the carbon number 6 that sticks up out the side of the ring, that isn't in the ring. So the 6 here will link to the carbon 1 of another glucose molecule. So basically this just means that a side branch will be started. So normally they link 1 to 4, so we'll have the 1 here, and then the 4 of the next molecule, and the 1. Alright, if we start one here with 6, this is carbon number 6 on this sugar, and the next molecule will have carbon number 1 sticking to it. And then that side chain can continue with 1 to 4 linkages in a straight line, and eventually it can branch again. So amylopectin, it's not that you only have 1 to 6 linkages. It's actually mainly 1 to 4, mainly straight chains, but occasionally you get these branches or these side linkage that comes off to one side. So you could see here, you can imagine we've got a sugar molecule here to the left and one here. We could still add one over here, a 1,4 linkage, so carbon 1 could link to a 4 of another one. But we could also have a side branch starting off to the side here. From carbon 6, we could link a sugar here, branching off to the side or upward. So, starch grains are basically little granules that can build up in chloroplasts or other storage organs. So, for instance, potatoes or the seeds, basically any part of a plant that needs to store, have a store of energy, or functions essentially to store any spare or excess energy. Seeds, you can see the reason they obviously need to store energy for germination. They won't get any energy until they actually start producing green leaves that can absorb sunlight. So these starch grains are actually very easily visible under the microscope. And so you can take a potato and rub it on a microscope slide, put a bit of iodine on it, chuck it on the microscope slide, and you should see lots of these black sort of little spots. And that's because iodine stains black. Starch stains black with iodine. So guys, starch is relatively insoluble, which means doesn't dissolve. Now it's not completely insoluble, but it's less soluble than sugar. And that's important because, like I said, if we had the equivalent amount of sugar, that would disrupt the cell's osmotic balance, the flow of water in and out of the cell. Basically, it would be too concentrated sugar. It would be like a syrupy kind of solution inside the cell, and that would be toxic to the cell. So it's important to kind of remove that sugar. Now, we don't want to just excrete it or get rid of it because it's a very valuable energy source. So the cells store it as starch, something that's a bit less soluble. Okay, so glycogen. Glycogen is basically how we store these sugar molecules. So glycogen is how animals store your sugars, so your mono and disaccharides, as essentially larger chains. So this is how we store some of our energy. Now, I should qualify that. We actually store most of our energy as fat, but that's kind of a more long-term store. So when you eat a meal, you'll break it down and often convert a lot of it into sugar. That glucose will circulate through the blood, and if you've got excess, it'll basically be taken up and initially stored as glycogen. Now, if your glycogen store is full, then it'll start making fat. But your glycogen is sort of more that temporary store, so that one actually fluctuates. And if you haven't eaten for several hours, you'll start to deplete the glycogen store, and when that runs out, you'll start breaking down your fat. So this is how we store those simple sugars as chains. So glycogen is actually very similar to amylopectin. It is a straight chain with 1,4 linkages, so made of alpha glucose chain with 1,4 linkages. And again, it's also got those 1,6 branches. So here you can see the molecule we're looking at the moment. We've got number 1 here, number 4. All right, carbon number 1, number 4. So these are the 1,4 linkages, giving us a straight line. And we might have a side branch, carbon number 6, sticking up the top here. We might have some glucoses linking off to the side here, probably in a 1,4 chain in a straight line for a while, and then at some point they'll branch again, another 1,6 linkage. So whenever we link the molecules 1,4, so link carbon 1 to carbon 4 with a glycosidic bond, these oxygens that bridge it, we get straight chains. And then whenever there's a 1 to 6 bond, we basically start a side chain or a branch that usually continues on in a 1 to 4 linkage in a straight line for a while before eventually it might branch again. Okay, so what's the difference between amylopectin and glycogen? Basically, glycogen just tends to have more branches than starch. It's a slightly more branch structure. Glycogen forms granules in our muscles and liver, and again, just to act as an energy reserve. So, lastly, cellulose. Alright, cellulose is the most abundant molecule on the planet. Organic molecule. So that's sort of what, the most abundant carbon-based molecule on the planet. And there's several reasons for that. Okay, cellulose is a very tough material. It's hard to break down. So when we eat cellulose, we can't really break it down, so it just passes straight through us, and that's why we call it fiber rather than. It's often why it's not referred to as a carbohydrate, but rather a dietitian would call it fiber rather than. It is actually still technically a carbohydrate. Okay, it's still made out of sugars, so it's a carbohydrate, and it's still got that general formula of the CH2O. But the difference here is that cellulose is made out of beta glucose. All right, so beta glucose was just the difference there. Is the orientation of those OH groups. So remember, beta glucose is the one where it alternates. It goes up from carbon 1. We've got an OH coming up, and then from carbon 2 down, up, down, up. It just alternates. Whereas before we were using alpha glucose to make our starch and glycogen, and the alpha glucose went down from carbon 1, down, down, up, down, and then alternating. Alright, so the only difference is the OH group on that carbon 1. Alright, so for beta glucose it sticks up. But this actually means that when we link our beta glucoses together, alright, they have to actually link. One has to be upside down relative to the other one. So imagine if you flipped this around, flipped the top and the bottom. Imagine they're connected here at either side, but we could flip it over by pulling down the bottom so it would rotate around the points at each side here. Alright, so just imagine we flip that over. That's how the glucoses have to connect. One's the right way up, the next one's upside down, the next one's the right way up again. So they actually have to be, they have to bond at 180 degrees relative to each other. Alright, the alternating pattern of this one molecule being upside down relative to the other maximizes hydrogen bonding within, but especially between cellulose molecules. So if we look at the carbon chain, if you imagine cellulose a chain of glucose like this, alright, one connected to the other, one of them relative to the other is upside down, so we've swapped. Imagine the writing here would be upside down. We swap the top and the bottom. What that means is that the hydrogen bonding occurs between all the OH groups. So at each point on the around the the ring here, remember we've got OHs alternating in an up-down pattern. So at each carbon, so one is no longer has an OH because that's where we've got that glycosidic bond formed. But here we would have an OH group that comes down, and then one that comes up. All right, and then one that comes here we would have had one that was down, and then one that comes up off that side chain. So that alternating pattern of OHs is what actually causes the hydrogen bonding. So OH groups can hydrogen bond, and the alternating pattern just means that they can pack together or come a little bit closer together, which maximizes or increases the attractive force of that bond. So the OH groups within a molecule are attracted towards each other, but also especially between molecules. So what I mean by that is that the OH group on this, if we had another chain up here, is attracted to the OH group on the neighboring chain. So we can lay these chains side by side to get that attractive bond. So that's actually what happens. We get about 60 to 70 cellulose molecules, so just a long chain of beta-glucose, which are attracted to each other. So they lay, imagine taking 60 or 70 cotton threads and lying them side by side. They'd be attracted to each other by hydrogen bonding between the OH groups. And so that forms what we call a microfibril. A microfibril is just a sort of almost like a tiny fiber, microfiber fibril. It's got about 10 nanometer diameter. You don't really need to remember that, but it's about 60 to 70 cellulose molecules kind of packed side by side together. And I'll show you a picture in a minute. So if we take a whole bunch of those microfibrils, they're held together in bundles called fibers. So imagine like taking cotton threads and taking 60 to 70 of them and kind of gluing them together. That's the hydrogen bonding holding them together. And then imagine taking bundles of those and tying those together. You're essentially what you'd be doing is making a rope. So the fibers again are held together again just by hydrogen bonding between those OH groups. So the reason it's so abundant, why this molecule is so abundant, is that plant cell walls, 20 to 40% of the cell wall is cellulose. So the most abundant molecule in living organisms is water, but the most abundant organic carbon-based molecule is actually the cell walls of plants. And because plants are the base of the food chain, we've got more plants or organisms that photosynthesize and that have cell walls than any other type. So that's why we have so much of this material. The other reason we have so much is that it doesn't break down very easily, which means when these organisms die, it tends to hang around for a while, whereas some of the other molecules are broken down a bit quicker. So here's what we were talking about when I was talking about those fibrils and those fibers. So you can see here glucose molecules linked in a long chain. And they'll get 60 or 70 of those sitting side by side with hydrogen bonding between, the OH groups on those chains. They bundle together into, so these long chains of beta glucose bundle together in what we call a fibril. Those fibrils bundle in together. This yellow thing here is a fiber. And those fibers, there are a few other polysaccharides. Now you don't need to know about those ones, but there's a few other polysaccharides that actually link the fibers together. So other polysaccharides help to cross-link or join those fibers together, and so what you end up with is they kind of act as a glue which holds those fibers together, and again just strengthens it strengthens them like a rope. So if we look here, the cell wall of plants actually looks like this. You can see the yellow rod kind of things here are those cellulose fibers. So each one of those made out of several microfibrils, and each microfibril made out of 60 to 70 cellulose molecules, so beta-glucose chains that are packed together or hydrogen bonded together side by side. A few other polysaccharides, things like hemicellulose and pectin, are the ones that help to cross-link it. So guys, cellulose fibers have a very high tensile strength. Tensile strength means that you can stretch them or pull them. So we talk about, in engineering, you talk about tensile and compression strength. Tensile strength is the force you can apply in a stretch when you try to pull something apart. Compression strength is the force that something can sustain or withhold without collapsing when it's squashed. So tensile being a stretch strength, and compression being a squash strength. So cellulose fibers have a very high tensile strength. If you try to pull on them, they don't break. So they make a mesh-like structure, cross-linked by those other molecules, and that helps give the cell its shape and structural rigidity, as well as limiting the cell volume. So imagine this is kind of like, pantyhose is kind of a network. If we take some pantyhose and try to blow up a balloon inside it, okay, it would limit the balloon. You'd probably struggle to pop the balloon if you were blowing it up inside like a pair of pantyhose. Or even if you imagine taking a net or like chicken wire and making kind of a little bit of a cage and trying to blow up a balloon inside that, assuming it's not spiky and it doesn't pop the balloon, it would limit the volume. There'd be a limit to how big the balloon can get. And that's an important function of the cell wall. But despite its strength, another really important feature is it's still fully permeable. So like the pantyhose or like the chicken wire, there's still enough gaps in there that things like water and other molecules can move through the gaps. So the cell wall gives that really strong structural strength, all right, without blocking out the ability of things to move in and out. And lastly, that tensile strength is really something, it's not to be underestimated. The tensile strength of these cellulose fibers is actually approaching or similar to the tensile strength of steel, steel cable or steel wire. So it's a very effective way of producing a strong cell wall. And it's the strength of that cell wall that, gives trees, in their trunks and things, they have thickened cell walls that gives the trunk of those trees that extra strength. And the amazing thing about these biological systems is, when we build structures and we need to reinforce it, we just make them bigger and add sections here and there. Whereas in a plant, it can really look at where is the largest force being applied, and it can reinforce that at the cellular level. So if one cell is having a greater force applied to it, it can reinforce the cell wall of that cell. All right, and it can even reinforce the cell wall on certain sides, not just on all sides. So you can really reinforce the structure where it needs reinforcing. And the great thing about that is it means that you don't have to waste any energy, you don't have to add extra weight, all right, by reinforcing parts that don't really need reinforcing. Really at that cellular level, at that kind of detail, you can reinforce the areas that need additional strength, or where there's strain or force is being applied, and areas where there's less force being applied, well, you can actually reduce the thickness of the cell walls and the rigidity, and thereby also the weight. So that makes trees have an incredibly efficient structure, very lightweight yet strong material, and that's why we still use it today to build houses and all those sorts of things. So guys, those are basically our polysaccharides. So starch was a long chain of glucose, was made out of amylose and amylopectin. Amylose just being a long chain of alpha glucose linked with 1 to 4 bonds. All right, amylopectin, another component of starch, being the same thing, glucose, alpha glucose linked with those 1 to 4 bonds, but with some 1 to 6 side branches. Glycogen, the way we store it, again, is just the alpha glucose linked 1 to 4 with those 1,6 side branches. So you can see here the 6 should actually be linked to the 1, all right? But we can see some side branches here. Glycogen, the only difference is it's basically the same as the amylopectin, except that it's often slightly more branched. And cellulose is just a straight chain, so no side branches, just a straight chain, except using beta glucose, which means that each molecule alternates. You can see this one, you've got the 6 coming up here, the 6, that bit that sticks out of the molecule coming down. So they've rotated 180 degrees. We're alternating. One glucose molecule is the right way up, the other one is upside down. So that makes these cellulose molecules, these long chains, pack together or hydrogen bond more efficiently. So they pack together quite strongly and basically form these fibers that make up the cell wall. So guys, I hope that helped. That's it for polysaccharides, and we'll get on to one of our other biological molecules in the next video.